syntax as a fingerprint of human language. Why, you may ask, should syntax be so important? Well, syntax, you called it grammar in school, is the backbone and core of every language. And every child learns every language in the world, and we speak and listen automatically. However, usually we are not able to declare the rules according to which we build the sentences. So the question is, so what is the brain basis for all of this? And we will allude to this today. So there are several animals, that is animals like parrots and dogs, and they can learn words, but they cannot acquire syntax. And even our closest relatives, the chimpanzee, they differ from us in their ability to process syntax. This case study may illustrate this best. So the best example may be this case study. Here, what you see is the mean length of utterance of a hearing child. And you can see it starts off with 2 words and then brings more and more words together to come up to a sentence. Now look at a deaf child. This child learned sign language, though it started somewhat later, but then piece by piece, and then dramatically increased the number of words in an utterance. The chimpanzee, however, even after extensive training, does not reach never this level. So what we can conclude from this is that non-human primates can learn words, but they do not combine those into sentences. So the crucial question then is, what is the brain basis for the human capacity of language? There are 2 major regions that have been identified already decades ago, Broca's area and Wernicke's area. Broca's area was thought to support language production. These days, we know that there are 2 areas. One is 44, one 45. They are cytoarchitectonically differently, and they also are thought to support different processes, syntax and semantics, respectively. There's this huge area in the temporal cortex, Wernicke's area, which at least these days is thought to hold the mental lexicon. In addition to that, there are fiber tracks, white matter fiber tracks that connect those 2 brain regions. And these fiber tracks are dorsally located or ventrally located, and they will play a major role in the talk during today. So let's see, when we compare the human brain to the non-human brain, are there differences? Yes, there are. So with respect to the cytoarchitectonic asymmetry of Broca's area, what we find is that this area is larger in the left hemisphere, which is the language dominant hemisphere, than in the right hemisphere. And the developmental trajectory of those 2 regions with respect to their asymmetry is different. For the area 45, which possibly supports semantic, this asymmetry is already reached by the age of 5 years. However, area 44, the area that supposedly supports syntax, the asymmetry is only reached by the age of 11 years, so much later. When comparing this to the chimpanzee, there is no asymmetry whatsoever, even in the adult brain. When looking at the fiber tracks, we also see interesting differences. In the human, there is this dorsal fiber tract, color-coded in purple here, and the ventral fiber tract, color-coded in green. And the dorsal fiber tract is much stronger than the ventral fiber tract. In the macaque monkey, however, the reverse is true. So you see a weak dorsal fiber tract and a strong ventral fiber tract. Now the question comes, what is then really the behavioral difference once we do a fair test between the human and the non-human primate? And language as such may not be a fair test. So researchers created artificial grammars that should also be possible for non-human primates maybe to process. So a simple grammar is a finite state grammar where, for example, every A element is followed by an E element. And you see the syllables here. Phrase structure grammars are more complex and have an embedded structure. So these two grammar types were tested in humans and in tamarin monkeys. For humans, it was pretty clear both types of grammars were learned after very few sentence exposures or syllable exposures. However, the tamarin monkey, they only learned the simple grammar and they never learned the complex grammar, even after intensive training. So from this, we can conclude that complex sequences, even in this artificial grammar sequences, can only be learned by humans and not by non-human primates. At this point, the question arose, given this difference of the two grammars, is it also that the two grammars are processed by different areas in the human brain or by the same area? In order to test this, we took the very same grammars and presented them to humans during an fMRI experiment where we image the activation of the brain. So what we found was the following. The simple, the finite state grammar activated the frontal operculum. The frontal operculum, but not Broca's area. And this is interesting because the frontal operculum is a phylogenetically older cortex than Broca's area. However, then when looking at the phrase structure grammar, we also see the frontal operculum, but now Broca's area, that is 44, comes into play. So what this means then is that the structure hierarchy in phrase structure grammar, at least in these artificial grammars, is processed by Broca's area 44. We also had the possibility to look at the fiber tracks in those very same subjects. And when seeding in the activation spot of each of the subjects for the simple grammar, namely the frontal operculum, we found a ventral fiber track as displayed here for the 4 subjects. We found that in all the subjects. Now look at the other grammar where we had activation in 44. Once we see it in 44 and ask, where does the fiber track bring us? We see the dorsal fiber track. And this is also true for all the subjects. So it appears that there are interesting differences between the sort of human and non-human brain, but also for the two grammars with respect to the older cortex and the younger cortex. However, now the question may be, is that then really true, what you have shown there with the artificial grammar for natural grammar? And German is a wonderful language where you have center embedded sentences, which, brings you the same embedded structure as for the artificial grammar. So here you see the relation between the subject and the verb for the different sentences and sub-sentences built up this embedded structure. We also had longer sentences where we had no embedding in order to see whether our effects are really sort of separating between the distance between those elements or the embedding. And as expected, we found activation in Broca's area. The orange is the main effect, that is the activity for the syntactic hierarchy. And this is placed in the cytoarchitectonic defined area, color-coded in green here, 44, not in 45. The distance effect is located in the left inferior frontal sulcus, that is color-coded in blue here. Usually these areas work together when it comes to process those complex sentences. However, what you really want to show is that when you look at Broca's area and you make the sentence more and more and more complex, does this really increase its activity? And this is, what we also were able to show with respect to a German sentence, where you either have a simple structure, a canonical structure, where the subject is in front and then come the two objects in a sentence like, today the grandfather, the boy, has given the lollipop. And so what you can do in German is the following. You can bring an object in front of the subject, making a medium complexity. The sentence doesn't get longer, just more complex. And you can do even worse. You can bring the to in front of the subject and then reach a high complexity. So now looking at Broca's area and the activation in that area, what we see is that as the complexity increases, the activation increases in this area from low to medium to high. And with this, I think we can say BA44 is responsible for the processing of syntactic complexity. With this, we can even come to a conclusion here, namely that at least for the adult brain, we see that the function for syntactically complex sentences is going up and activating the Broca's area. Other studies, and also our own, have shown that in addition, you also activate the posterior temporal cortex. These two regions are connected by the dorsal fiber tract. But at this point, I have to say that we cannot be sure that this fiber tract is really supporting complex syntactic processes, because the imaging of the white matter does not give you any indication about the function. So you have to use another way to test this. And I will come back to that later. Before I do so, however, I would like to ask an interesting question, at least for those that speak other languages or are grown up in other languages. Is the language network universal, or is it specific for each language? And so we looked at different brains from people who had monolingually learned either German or English or Chinese. And when we bunch this together, all the data that we have from those three groups, you see the known language network with its dorsal and ventral fiber tract. However, a detailed analysis of the strengths of the fiber tract in each of the three groups, German, English, and Chinese, shows us that there are interesting differences between the strengths of the fiber tract. I allude here to the dorsal fiber tract, which is stronger in German and in Chinese and weaker in English. And this makes sense because English is a fixed word order language with less morphosyntactic elements than, for example, German. So from this, we can conclude, yes, there is a universal language network, which is modulated by the lifelong use, however, of a particular language. So far, so good for the adults. And so the adult network, here we see it again. And also, I would like to point out that here the dorsal fiber tract has a blue part and a purple part. And this is interesting once we compare the adult brain to the infant brain. And so the question here was, is what we see for the adults with respect to the white meta-language network already established at birth in the pre-linguistic child? And we had the ability to analyze the white matter fiber tracts in newborns. So the comparison between the newborn brain and the adult brain revealed some interesting differences. In the adult, we have two dorsal fiber tracts, one going to Broca's area, that's the purple one, and one only going to the premotor cortex, not reaching out that far into the inferior frontal cortex. This is color-coded in blue. And also you see the ventral tract, which is already pretty well developed in the newborns. Given that the way that we do our analysis, we capture, among others, the myelin of those fiber tracts, I think it's important to note here this difference, because once the myelin is there, the information transfer from A to B, from one region to the next region, is much better and faster. So with respect to the dorsal fiber tract to Broca's area, we can say that at least at birth, it is not myelinated yet. So actually, it's not functional in the sense that it's functional in the adults. Now, what can those infants do with this reduced language network? We reasoned that the pathway to premotor cortex should allow phonologically based learning. We tested this in 4-month-old infants. And those 4-month-old infants, they had to learn Italian. Actually, they had to learn the dependency between the auxiliary and the ending. For example, sta, ando, or are. We used more than 30 verbs in our tests. And so the children really had to abstract from the verb stem and learn the regularity. We also had incorrect sentences where this relation was not correct, because we wanted to see, do our 4-month-old infants learn the difference between the correct and the incorrect ones? So in our tests, what we did, we had a short learning phase and then a test phase and a learning phase again, and so on. And in the learning phase, the infants only heard correct sentences, and the test phase, they heard correct and incorrect sentences. We recorded the electrophysiological response in those 4-month-old infants and found the following. In test phase 1, there was no difference between the correct and the incorrect endings. So correct blue, incorrect red. However, in test phase 4, you see this dramatic difference between the correct and the incorrect sentences, showing that infants were able to learn this. And you can also see that this activation, is widely distributed across the brain. So from this, we can at least conclude that the detection of the phonological dependencies is there. And we take that this is a prerequisite for learning syntactic dependencies, because these often go together. Now, however, the way up to learning complex sentences is really a long one, from 4-month-old infants then to older children. So what we know from a couple of studies that have been done over and over in different laboratories is the following. Children deal well with very simple sentences like the dog beefed the lion. Don't ask yourself what beefing means. It's a non-word. So we didn't want to sort of bring in semantics here more than just necessary. However, when you now look at sentences where the object is in front, so object first sentences, you see that even 3-year-old or 2.5-year-old and 4-year-old children are not performing above chance. So performing here means they have to listen to a sentence and the choice of a picture, which is either the correct match to the sentence or not. Only by the age of 7 are they just coming above chance for those sentences, which are not long, they are just complex. So given that we have already made the statement that the dorsal fiber tract connecting to BA44 and the temporal cortex is crucially involved in the processing of syntactic complexity, we now reasoned the following way. If so, then the dorsal pathway may not be fully developed in children. So now we had to look at the whole development. And we did this across development, children between 3 and 10 years, and also looking at adults. And the dorsal fiber tract, color coded again here in purple, you can only see it with a blanket eye that this is much stronger in adults. And the ventral fiber tract is pretty much established already. So when looking at the differences more carefully, we see that the strength of the dorsal fiber tract increases from age group to age group. More importantly, however, this increase predicts and is correlated with the children's performance on processing complex sentences. And this significant correlation you can see here. So with that, we can conclude that the dorsal fiber tract targeting 44 is crucial for the processing of syntactically sort of complex sentences. So where does all this brings us with respect to our question, namely the syntax as the fingerprint of human language? Well, we have just learned from newborns to adults, there is a long way, and it is related to the maturation of the dorsal fiber tract. And when we look into the phylogeny, we also see that the dorsal fiber tract here plays a major role in the ability to deal with syntax. So with this then, I think we can say that the strength of the dorsal fiber tract 244 appears to be crucial for the human capacity of syntax. And what this means is that syntax as a human unique trait is maybe grounded in the connectomics of the human brain.